Hey everybody, today we are going to be talking about Punnett squares. You will need a pencil to write with and then 2 additional colored writing utensils. So when we talk about Punnett squares, a Punnett square is going to show us the probability that an offspring will have a specific trait. So if we take a look up here, we are going to be talking about plant height, and what we're going to be focusing on are the alleles for that. The alleles could possibly be T, which represents, or capital T, which represents tall, or lowercase t, which represents short. Now, in order for us to take this information and figure out how to set up our Punnett square, we first need to make a key. So a key is going to show us several things. So make sure that you guys are writing this down as well. A key is going to show us our genotypes. And the genotypes are going to be represented by letters. Then the genotypes will tell us our phenotypes, or what it looks like. So what we're going to do is we are going to take those 2 alleles, the capital T and the lowercase t, and look at all the possible allele combinations we could have. Now, when we talk about allele combinations, we're going to be looking at 3 different types. The first is going to be a homozygous dominant. That means capital T, capital T. The second type would be a heterozygous, which is capital T and lowercase t. And the final is going to be homozygous recessive. So homozygous, same, recessive, lowercase letters. So that means little t, little t. So these are the 3 possible genotype combinations we could have. Now, the phenotype combinations are the different types of physical characteristics. So that means big T, big T. Big T, big T would look tall. I know this because I can use my key up top here to help me figure that out. If a capital letter is present, it will always be the dominant trait. So capital T, lowercase t, again, there's that capital letter present, so it's going to be tall. That dominant trait overpowers the recessive trait. And finally, little t, little t. This allele combination is going to represent short. So my phenotype for little t will be short. Now, we're going to take that this genotype, phenotype key information, and we'll be using that later on as a reference. Now, we need to determine the parents' genotypes. Again, genotypes, as we wrote down, are letters. And that was given to us in this top part here. It says cross a heterozygous and a heterozygous plant. Now, this is where I'm going to be using my 2 different colors. I'm going to be using red and a bluish purple. So my first parent, or parent 1, is the one that's listed first. So for parent 1, it's going to be big T, little t, or heterozygous. My second parent is also heterozygous, big T, little t. Now, when we take this information, we will be plugging it into our Punnett square. Now, sometimes on Punnett squares, we like to extend that line out. We like to extend that line out so we know that we need to separate our alleles. Now, in middle school, you might have also been taught that a certain parent goes on the top or the side, or certain gender goes on the top or side. It doesn't matter. What we are mainly focusing on are the inside. So whatever's on the outside, whatever order that goes in, that's fine. Normally, we will take parent 1 and put parent 1 on the top. So I'm going to separate that allele combination. So capital T, lowercase t. I'm then going to take parent 2 and put them on the side. Again, separating those alleles, capital T and lowercase t. Now, before I go forward, I'm going to double check. Okay, my parent 1 is good, parent 2 is good. Parent 1, parent 2. Now, what we need to do is we need to now combine these separate alleles. So our dad's first allele, that is going to drop down, and mom's allele is going to move over. Now, I like to read this like I'm reading a book, so I'm going to start on the left and move my way over. So 1 moves over, mom's capital T moves over, dad's capital T moves down. Now, if we move to the second box, our capital T from over here is also going to move over, and that lowercase t is going to drop. Now, if you noticed, I did put that capital T first. That capital T is a dominant trait, so that dominant trait always comes before the recessive. You can also think of it kind of like your name. If I was to write my name with a lowercase w, capital E-I-S, that looks super weird. So we always start off with a capital letter. Now I'm going to go down to my next line. I'm going to start at the left. My capital T will drop. My lowercase t will slide. Now, when we get to this last box, we're dealing with 2 small t's. It doesn't matter what order they go in. So you guys can put whichever color you would like first. Doesn't matter. Same with this box over here, okay? If they are the same height, if they are homozygous, it doesn't matter what order they go in. So now we are going to be looking at our potential offspring. So this does not mean that our parent is going to have 4 children. What this means is if they have a child, what is the chance that they are going to have a certain trait? So when we are at this F1 generation right here, we are focusing on what is happening on the inside. So we are focusing on these boxes here. So as a reminder, genotype, we're going to write down letters. And we're going to go in the same order as we did our key. So big T, we're going to start with homozygous dominant. And if I look, I'm going to start here at the left and I'm going to work my way through. Big T. I have 1 genotype that is big T. And I'm just going to put a little check mark in the corner as a reminder for myself that I did that. My second one is big T little t. And I've got a big T little t here and a big T little t here. So that's 2. Big T little t. And finally, little t, that is right there. I've got 1 little t. Now, if I can, you guys don't have to do these little check marks like I did. They're just my reminder. So I can look back and say, okay, I identified each box. They are accounted for. You can also look and see 1 plus 2 plus 1 equals 4 because I have 4 boxes. The next is going to be phenotype ratio. Phenotype is what it looks like. Okay, we already figured out what they look like. They're either going to be tall or short. So I'm going to write that here. Tall and short. Okay, now for tall, I can come right up here and see, okay, well, I know that big T and big T little t mean tall. So 1 plus 2, that gives me 3. And little t means short, and I only have 1 of them. Again, this should equal 4. Now, we're going to move down to the next example. If you need to rewind or do anything for this top one, please do so, but we're going to move on down to this bottom one. What I recommend you guys do at this point is pause this, and I want you guys to make a key and identify the genotypes of the parents, and then come back to see if you are right. Do not go any further until you do so. So the first thing that we are going to do is we're going to underline some information. So the first piece of information, it says in pea plants, green seeds, capital G, are dominant. And then yellow seeds would be lowercase g. So we can put a capital G, lowercase g, or just underline it. So I'm going to take that information first to make my key. Again, I'm going to identify my 3 types of genotype combinations, or my 3 type of allele combinations. And I always go in this order. I think of it as biggest to smallest. So we have our homozygous dominant, our heterozygous, and then our homozygous recessive. We know that these 2, because they have that dominant trait, and it says green seeds, capital G, are dominant. So that means these 2 allele combinations are going to be green. The bottom one, little g, These will be yellow. So that's my first step. Then I need to look and find my parents. So it says, show the genetic cross. This is like your key word right here. When it's talking about a cross, this is going to lead us to our parents. So it says between a homozygous green plant and homozygous yellow plant. This will be our first parent because it's the first one they listed. This will be our second parent. Now, when I write things down, I like to think about homozygous versus heterozygous. Homozygous is same. My green plant is represented by a large G. Homozygous, same. Yellow is a little g. So now I'm going to take that information and I'm going to go to parent 1. Parent 1 says same, big G. So that means that I need to have both letters from my parent be the same, and they're both going to be capital G. Same, big G. Parent 2 says same, little g. So that means both parents are going to be small. I'm actually going to use my colors like I did before, just so that we can see that. Okay, so parent 1, parent 2. All right, so as long as this is correct on your paper, I want you guys to then pause and try to fill out your Punnett Square and get your ratios as well. Come back and check your work. So my first parent I'm going to take and put on the top. Again, you can extend your lines if this helps you and is a reminder. You don't have to, but it is an option. I'm going to put my first parent up top, separate those alleles out, and my second parent on the side, again, separating those alleles out. And then we're going to cross that information. So big G drops, little g slides. Big G drops, little g slides. Go to my next line. Big G drops, little g slides. Big G drops, little g slides. Yours should look like mine. Now for my genotype ratio, as we did above, I do need to list all 3 of my potential allele combinations out. So I'm going to come back here to my key, and I'm going to list all of them out. Big G, little, little, little. So I don't forget to write stuff down for them. So I'm going to go through. Do I have any big G's? Nope. So I'm going to put a 0. I'm going to put my little colon. Big G, little g. I'm going to go through. I have 1, 2, 3, 4. Perfect. And then finally, little g. All of my boxes are marked already, and that means I have 0 of little g or homozygous recessive. My phenotype ratio. I know that my 2 phenotypes are yellow and green. So I'm going to write out green and then yellow. Again, I'm going to go through. Green is either represented by homozygous dominant or heterozygous, and all 4 of mine are heterozygous. So that means I have 4 green and 0 yellow. Make sure yours looks like mine. All right, if you have any questions on this, please let myself or Ms. Fash know. What you guys are going to work on after this is you're going to flip to page 8, and you are going to be going through problems 1 through 5, so page 8 and 9. There is one thing I would like for you guys to take a look at, though. When it says show the cross between 2 heterozygous parents, and then this was your information for flower color, so that's the flower color. So it's saying that parent 1 is heterozygous and parent 2 is heterozygous. Both parents are heterozygous, and that's okay. Your parents can be the same, your parents can be different, but parent 1 is heterozygous. Parent 2 is heterozygous. So make sure you guys pay attention to that part. Again, you are going to work on pages 8 and 9, and if you have questions, please let us know. Thank you.